Good afternoon, everybody. I will start with a bit of presentation of myself because I don't think many of you knows me. So I'm a biologist by formation. I studied here in Trieste, but I soon became a virologist. I started to work with viruses during my PhD at the SCGB here in Padriciano. And then I moved for several years in Scotland to work in the University of Glasgow at the Centre for Virus Research. And as the name can suggest, is an institute fully dedicated to virology under many points of view. So after a long postdoc there that covered also a pandemic, a wonderful period for a virologist, I obtained my own funding and I came back when I joined the Area Science Park as a researcher, where I work in a strong collaboration with Alessandro Marcello at the ICGB. So I thought for this presentation to talk a bit about some topics that I touched during my previous year work, topics that I really enjoy, and I really hope to work again on them. And I think that this platform is the perfect place to get a lot of multidisciplinary insights. If I have time, because time is quite short, I will give just a brief description of the current project I'm working with Alessandro. So I will start with vaccine development because this is what brought me to Glasgow. I'm interested in RNA viruses, envelope RNA viruses, and when I'm talking about vaccine development, my interest is always in the humoral response, so basically antibodies. And to explain some important part of the vaccine development, I will touch a bit of background about aspects of virology. So forgive me for this tiny lesson of virology, may be very basic for some of you, completely new for others. So viral fusion is basically the fusion between the membrane of an envelope virus and the membrane of the cells to be infected. And this is an essential step for every envelope viruses in order to infect a new cell. It is performed by the fusion proteins that are viral glycoproteins that you can see the that basically they surround the structure of the virus. These proteins are responsible for interacting with the molecules on the surface of the cells to be infected, and going through many conformational changes, they mediate the interaction between the two membranes to generate a pore that allows the gene of the virus to go into the cells to be infected. So if this mechanism is not happening, the virus cannot infect the cells. At the moment, the viral glycoprotein, the fusion proteins, are divided into 3 classes, but probably there are many others that are still to be discovered. And as you can see here, we have class 1, class 2, class 3. The proteins goes through many conformational changes. There is all a maturation step that is prior to the interaction with the new cell to be infected, and then there is the process of fusion. What is interesting for us is in particular the native state here and here and here, because this is the fusion competent conformation state. The glycoproteins are metastable. They have to undergo conformational changes that can be triggered by many things, by proteolysis, so cellular protease that will cut the protein, interaction with co-receptor, like it happens for the spike of SARS-2 when it interacts with the ACE2 receptor, and pH changes. Everything happens because this fusion has these conformational changes needed for fusion must happen at the right moment, at the right place, not too early, not too late. But why this is important for vaccine design? And everything comes down to the immunogen structure. So basically, what do you want to give to your immune system in order to produce what you need to protect yourself? In our case, it's basically putting together the knowledge of all these pre- and post-fusion conformation structures together with the knowledge of where are the neutralizing epitopes. So where are located the portion of the protein recognized by antibodies that can neutralize the virus. We put together this knowledge in order to design the vaccine that will stimulate the production of neutralizing antibodies. And to do so, you want to basically stabilize the native state of your vaccine and avoid a premature folding of this protein to the post-fusion conformation. Because if you think that on the virus, all the proteins surrounding the structure are in the native state, so ready to fuse, forget about that. Unfortunately, you have a mixed population. You can have a native state, you can have immature proteins, so they didn't reach the native state, or you can have proteins that are a bit too quick into going in a post-fusion conformation. So I will give two quick examples of how these things are put into good use in designing a vaccine. The first is my personal work in Glasgow. We work on designing a vaccine against Zika virus. Zika virus, the glycoprotein is the envelope protein. It belongs to class 2 fusion proteins, and the native state is in form of dimer. As I said before, you can find this protein in immature state, immature, so native state, in post-fusion. All this contributes into making your immune system to produce a lot of antibodies that are not really neutralizing. What we did is to basically insert a mutation into the sequence of the envelope protein in order to generate a disulfide bridge here, where one envelope protein is facing the other envelope protein in the dimeric conformation. So our vaccine, our protein is forced to be in a dimeric conformation, and this basically lead to producing not more antibodies, but more neutralizing. So basically, we aim to quality over quantity of the antibodies. A second practical example that we are very familiar with are the vaccine against SARS-CoV-2. SARS-CoV-2 is a class 1 fusion protein, the spike. In this case, is in form of trimer, characteristic of class 1. And again, on the viral particle, it can easily fall back into a premature post-fusion conformation. And for instance, this was the case of the failure of one of the first Chinese vaccine, because during the inactivation process, they basically, the reagents that were used were pushing towards this post-fusion conformation, generating a vaccine that was not actually very useful. Many of the vaccines that were produced, for instance, Moderna, Pfizer, not AstraZeneca, interestingly, they decided to stabilize this native state by inserting 2 proline here. This is the sequence of the spike protein. And this is shown to stabilize a bit this trimeric conformation. Others, like Novavax and Johnson & Johnson, they even included another modification. So basically, they want to change a bit the polybasic cleavage side, that is where the furin can cut the spike during the maturation process. So basically, it's 2 different mutations with the same aim to stabilize the native conformation. And with this, I will move to the second topic, that is the impact of the emerging SARS-CoV-2 mutation on the spike plasticity. So when the pandemic started, I was in Glasgow, and I was enrolled into this Genotype to Phenotype UK National Virology Consortium that basically was created with the aim to study how all the emerging mutations in SARS-CoV-2 were affecting the virus regarding transmissibility, severity of the disease, effectiveness of the vaccines, and also of the treatment. It was a very big consortium. You can see that basically the major universities of the United Kingdom were part of it, and it was also very prolific in terms of studies. We made a lot of different kinds of experiments. We collected a lot of data. It would be very difficult here to summarize everything, but I will focus on one aspect that we found very interesting. So SARS-CoV-2 can enter in the cells following 2 different routes. Route 1, basically, it binds to the cell surface, of course, and mediates that fusion that I was talking about before at the cell surface level. Instead, route 2 is a bit slower. It's more a bumpy road, while route 1 is on a highway. Route 2, the virus goes into the endosome, and when it reaches the late endosome, here happens the fusion. So the fusion is happening in both cases, but in different locations, cell surface or the endosomal vesicle. What characterizes the 2 entry route is which protease, cellular protease, is used to cut the spike and allow these conformational changes. We have TMPRSS2 in route number 1 and cathepsin in route number 2. When Omicron appeared, at the beginning, the viruses were able to use both almost in an equal way. Then some variants evolved in order to prefer route number 1. So the Delta variant was extremely good in using route number 1, and that meant that it was also able to mediate cell-cell fusion a lot. But then at the end of 2021, Omicron emerged, and Omicron immediately showed to be a very different virus. It showed an extremely different behavior, in particular for the preference of the entry route. So it was poorly using entry route number 1 and mostly entry route number 2. This is something that we observe, and also many other colleagues. So going back to the study we performed, again, a lot of findings, but the one I want to focus now is the fact that we found out that other variants before Omicron were showing a preference for other variants were showing to prefer entry route number 2 prior to Omicron, but they were never noticed because they didn't share a lot of other characteristics of Omicron. And we saw that also this preference for an endosomal entry route didn't correlate with the decrease of lethality in animal model that was instead seen for Omicron. The other observations were more about the evolution of the Omicron sublineages. So all this led to a lot of open questions. È diverso forse? Non c'è. No, è diverso. Mi dispiace. Okay. A lot of open questions. The one I want to focus on is why some variants are showing this preference for endosomal entry route. Why they are not using the quick TM/2 entry and instead are going for a much more complicated endosomal entry. What we think is that it's a matter of trimer stability. For any reason, the trimer is more or less stable, and it is basically the site of cleavage for TM/2 is less accessible. We are still trying to find out. Maybe the virus is stalling on the cell surface until it is taken by the endosomal entry route. We don't know, but this may have implication in helping designing the next generation of vaccine. The fact is that it's not easy to give an answer to this. And this is a representation of all the mutations in the variants we study. So these are the variants. This is the sequence. Well, these are the residues. And you can see that the mutation. So it's very difficult to find two variants with the same phenotype mapping down to the same mutation. It's not so easy. Maybe it makes a bit more sense if we look at the location of the mutation in a more 3D structure, but still it's a very complicated puzzle. And what I really hope is that deep learning can help entangle this complicated puzzle. Then I will touch the third topic that I developed a kind of interest in the past years, and is the application of antiviral peptides. I don't think I'm the best person to give an introduction on antiviral peptide, because I think that we will have a speaker in the future from the Crystallography Institute that will probably talk much better than how I'm doing. Anyway, antiviral peptides are a subcategory of antimicrobial peptides. Is an active field of research. This short sequence of amino acid can actually have an antiviral activity. According to many modes of action, they can destroy the structure of the virus. In this case, they are defined virucidal peptides. They can interfere with the viral binding, or they can interfere with the replication, or they can even modulate the immune response. Of course, there are a lot of problems into working with antiviral peptides. They are not exactly cheap. There are issues with the ADME, absorption, distribution, metabolism, and elimination. And you may need to perform some modification in order to improve the performance. Anyway, if I'm interested in antiviral peptide, is because of Mario Mardi-Rossia, who is a researcher here at University of Trieste. He is working on antimicrobial peptide for a long time. And when you want to design or discover antiviral peptides, there are different, I think I'm not teaching anything new to you, but there are different approaches. You can use a computational source, or you can take from the nature. This is what Mario and his colleagues did. They took antiviral peptides from, antimicrobial peptides from different animal sources, and they focused the research on proline-rich peptides. So Mario came to me, say, I have a library of antimicrobial peptides. Do you want to see if they have activity against viruses? I said, well, why not? So I tested them against Zika, SARS-CoV-2, and yellow fever. Listening to the last seminar, I realized that our work of validation of drugs is quite different from the approach that structural experts are using. So it was very interesting. Anyway, what I did was to test all the drugs and see that some of them were able indeed to block replication of Zika virus. It was just a generic test because this is the representation of all the steps of a viral cycle. What you usually do at the beginning is just to add your drug or your peptide in this case, and then come with the virus, maintaining the peptide. So you're covering all these steps of the viral cycle. Once I found some positive hints, I started to go a bit deeper. So you can play with the timing when you add the drug or the antiviral peptides, and you can try to discriminate to what is working at the pre-entry level and what instead is working at the post-entry replication level. So this is what I did. Here you can see the infectivity, so how much the virus is able to work, no peptides. In both cases, here I wrote virucidal, I meant pre-entry, sorry. In both cases, of course, there is nothing there to block the virus is growing. You can see that almost none of the positive peptides is blocking the replication at the pre-entry level. Instead, all of them are acting at a post-entry level. So they are interfering with the replication of the virus. At this moment, I didn't have any more time to play with these antiviral peptides. If you ask me what was my bet, was that they were interfering with some of the viral enzyme, like the protease or the polymerase. Why I'm saying so? Oh, at least they were interfering with viral proteins. I'm saying so because when I screened against SARS-CoV-2, there was no activity at all. So probably it's not messing with some cellular component. Instead, a very preliminary experiment on yellow fever, that is a relative of Zika virus, both flaviviruses, is showing efficacy too. So probably there is a common target. And inspired by the very first seminar, I reached out to Francesca Cuturello from the Laboratory of Data Engineering, and I asked her for help if she could try to see if this peptide were actually binding to. And this is what she's doing right now. She's using AlphaFold fine-tuning for this kind of prediction. And I think that something is coming out from this. And the last part is the application. Of course, the immediate thought is clinical application. Here, I want to focus on something a bit more particular. It's probably a niche. It can be applied only to arboviruses, so viruses that are carried by arthropods, by insects. And this is a collaboration with Maria Vittoria Mancini. She is an entomologist at University of Pavia. And this application is paratransgenesis. I don't think you have never heard about that. Basically, this entails the use of genetically modified insect symbiotes. So bacteria or viruses that can infect insects, but not men, neither woman. And they can basically express, so you engineer these symbionts in order to express the molecule that you want to use as an antiviral. So this molecule will be expressed directly inside the vector, the mosquito in our case, and they will block the replication of the virus at the insect level, so before it reaches the human population. What you do is basically you perform all your antiviral peptide discovery process and validation. Then you optimize your peptide in order to be able to be expressed in this system. So you create your expression plasmid, you pick the suitable symbiont. In our case, we will work with bacteria, and you transform these symbionts. Then you use the transformed symbionts to infect the insects that you are aiming to, in our case, Aedes aegypti, for instance, and you will have a paratransgenic population. At this point, you need to check that the symbiont is distributed in the body of the insect, that is stable, so you have to pass multiple generations and check that the symbiont is still there, that maintains a good fitness, otherwise it will not compete with not transformed symbionts and it will be lost in the generation. And of course, it has to maintain the antiviral capacity. Only at this point, you can hope to test it on the field, like releasing the insects and see if they are decreasing the presence of the virus into the mosquito population. And briefly, the project I'm working at the moment with Alessandro. So this project is called antiviral stress, and the stress I'm talking about is the unfolded protein response. So the response mechanism that occurs when the endoplasmic reticulum is in a situation of stress. What Alessandro's laboratory saw some years ago is that if you take some cells infected with flavivirus, you can see that this response to the stress appears like 8 hours post-infection, and then after 16 hours, you start to see an interferon response, so the innate immune response, but none of this is able to block the replication of the virus. What they find out is that if briefly after infection, you supply the cells with a drug that can induce the UPR, both unfolded protein response and interferon response happen much earlier, and this can block the viral replication of the flaviviruses. So it seems like UPR and interferon response are working together in synergy. And this can open the road to identify potential antiviral drug that can act with a broad range on flaviviruses, not specifically acting on the virus, but more on the cellular pathway. So my project is focusing on understanding why the infection alone leads to activation of UPR and interferon response so late without this synergy. Why, if you add the drug earlier, the synergy is happening? And also, can this mechanism be applied to other virus families? And we pick, of course, coronaviruses. Unfortunately, just before I started my work, it was discovered that with SARS-CoV-2 infection, even if you add an UPR inducer, what you have is only a partial inhibition of the UPR or partial activation, and you still have the very messy interferon response that characterize SARS-CoV-2 infection, and none of these can block the replication of the virus. So now the project is a bit changed, and we're trying to understand how SARS-CoV-2 can partially block the activation of the unfolded protein response. And this is all